Hello, people. So I just uploaded the updated PowerPoint and the notes for lecture number 1. So if they weren't there in Moodle last time you looked, they're there now. So we got that out of the way. Did we go over the exam in the last class? Did we go over the essay questions with the exam? Blake, this is yes. This is no. Give me a yes or a no. Can't remember if we did that. Did we go over the exam last from Moodle? No? No, we didn't. Oh, okay. Thank you, Brian Pinello. Once again, you're saving my butt. All right, so let's go over the exam. Mainly what I want to do is just go over the essay questions because sometimes people tend to be more confused with those. The other ones are kind of like right or wrong. So I'm pulling my exam up here that we might talk about it. Study guys, exam, okay. Okay. It's gonna be okay, I promise. I tell you guys, this COVID stuff, I think I'm losing it. I do. I got nobody to talk to. It's just me. Okay, this is going to be a little weird because you're going to see it from my perspective rather than the way you usually see it, but I think we're going to be okay. We're going to be okay. Okay, so here, the first essay question. What would you predict to be the results from the following experiments? In vitro, a system of magnesium, GTP, and free tubulin monomers is established to promote treadmilling. When this is established, a solution of tau protein is added. What do you expect to see? Honestly, I got no idea. I just wanted to see that you were thinking about this and you had some intelligent hypotheses to come up with. So here's what I would expect. So you've got these things are treadmilling. You put the tau in there and it starts to like accumulate them together. So now you've got a bunch of microtubules bound together. But if they're depolymerizing at this end and polymerizing here, that tau protein is attached and it's going to make its way down to the bottom. When it gets to the bottom, is it going to stop depolymerization? If it does, then you'll just keep polymerizing and they'll get longer and longer. So you have long bundles. If it falls off the end, then not much is going to happen. They're just going to keep treadmilling. So could go either way. I don't know. I gave credit for either answer as long as you made some kind of an intelligent discussion of it. In a second experiment, tau and Taxol added to treadmilling microtubules. What would you expect to see in this experiment? Explain in detail. So tau is going to bundle them together. Taxol is going to stop them from depolymerizing. So you're not going to get treadmilling anymore because the minus end won't depolymerize, but the plus end should keep polymerizing. And so now you've got bundles of microtubules that are getting longer and longer. Eventually you'll run out of tubulin and it'll stop. But that's what I would expect to see. Okay, so that's the first question. Second question. By the way, people, remember, life is all about partial credit. Where would any of us be without partial credit? So if you don't know the answer, just say something. Talk about it. Talk about the process itself, which you can get right from the notes. And if you can't figure it out, you'll get 5 out of 10 points just for trying. Okay. Researchers who studied skeletal muscle contraction developed a model called the skinned muscle prep. In this prep, a single muscle fiber, i.e. one cell, is dissected away from its bundle. The fiber is put into a physiological solution and treated with detergents to remove all cell membranes, which means all the membranes in the cell, all of them. Given this prep, what would have to be added to the solution to cause the prep to contract? So a lot of people were saying things like acetylcholine to activate the motor end plate. Well, there's no motor end plate because there's no membrane. Some people were saying add an action potential to activate the T-tubules. Well, an action potential isn't something you can just add. You can add electrical stimulation, but there's no T-tubule to stimulate, so gone. There's no sarcoplasmic reticulum. It's a membrane, so it's gone. All there is open sarcomeres. What do you add to an open sarcomere to get it to contract? You need calcium to activate the actin thin filaments, right, to pull the tropomyosin off the myosin binding sites. And then myosin is going to need ATP in order to do the cross-bridge cycling. So that's all you really need, calcium and ATP, to cause that thing to contract. Okay. Bum, 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 bum. Skeletal versus smooth. Smooth operator. He's a smooth operator. I'm just killing time with my singing. I know it's not that good. Okay. Skeletal muscle can contract and relax in 100 milliseconds. Smooth muscle, on the other hand, contracts and relaxes on a minute's time scale. What is important about the two mechanisms that makes them different? Why is each important to the function of the tissue? Now, that last question, I didn't really grade based on that. I gave you extra points if you did talk about it, but in retrospect, it probably doesn't belong in this question because it's a very physiological question, not a cell biology question. So I didn't take points off if you didn't answer it. But anyway, what's the difference about these two mechanisms? Well, the skeletal muscle mechanism is very electrically connected, and electrical things are fast. So you've got the action potential comes down the T-tubule, and you've got a physical unplugging of the ryanodine receptor, and calcium floods out very quick. So really the only, let's see. So you got the electrical thing coming down, changes the channel, calcium comes flooding out, boom, you got contraction. And then when you close that ryanodine receptor, the calcium gets pumped back into the SR and it relaxes. So it's very quick. With smooth muscle, you activate it usually with a hormone, and then that goes through a G protein, which then activates another enzyme that produces IP3. I don't know if we talked about all this stuff, but then you get calcium released from the ER. Calcium binds to calmodulin. Calmodulin binds to myosin light chain kinase. And now you don't even have a sarcomere yet, not until myosin light chain kinase phosphorylates to myosin light chains, and then the myosin unfolds and it starts to bind to other myosins, and then you have to activate the thin filaments too. So there's an awful lot of steps between turning it on and it actually contracting, right? You got a lot of enzymatic steps, and every one takes time. So much fewer of them in the skeletal muscle than in the smooth muscle. Now, why is each important? Well, skeletal muscle has to contract fast, right? You have to react to the environment really quickly so you can change things. Smooth muscle, not so much. Smooth muscles, like your intestines contracting to push food along, not life and death. You're going to get there. And also with smooth muscle, it takes a lot less energy to contract than the skeletal muscle. So it's kind of conserving energy by not contracting faster than it has to. So that's more or less the physiological reason. Listen to me. Okay. That was the answer to that one. Cell crawling mutated cell line. This one really surprised me. Okay. Oh God. I did kind of hint that you might get a question like this. I think I gave a practice question like this at one point. You are a cell biologist studying the crawling behavior of fibroblasts. One of the cell lines that you're studying has one mutated gene. Okay, so there's one protein that's screwed up here. These cells are unable to move when stimulated. Okay, now we're going to look closer at them. You note that they can extend the lamellipodium, okay, which seems to be riffling. Now, I don't think I explained this, and I didn't take points off if you didn't understand it, but if it's riffling, that means it's treadmilling, okay? You've got polymerization at the front end of the lamellipodium, depolymerization at the end, and you're treadmilling. But is unable to move forward. What protein may be mutated? Why would you say this? Now, I would say a good third of you said, well, it must be RAC, because RAC's involved in making a lamellipodium. But it's got a lamellipodium. So if that's fine, then the RAC protein can't be screwed up, okay? So I understand how your mind went from lamellipodium to RAC, but you missed the point that, you know, RAC is bad, then you're not getting a lamellipodium. Anyway, what could be wrong? Well, it looks like it's not attaching to the surface if it's riffling, right? You're treadmilling, so you must not be going to the next step, which is attachment. So mainly what I was looking for was any kind of protein associated with the integrins and attachment of the lamellipodium to the substrate. But other people said Rho, which is involved in stress fibers. Sure, why not? I mean, it's not moving forward. Maybe Rho is not working and therefore that's not happening. Or maybe the myosin that's involved in smooth muscle cell crawling. Remember, there's a lot of myosins. The cell could be normal and still lacking this one protein. So that's a possibility too. So basically anything that would stop the cell from moving once it's already got its kind of body established there to move. Okay, what else? Do we need to go over these? These are kind of, no, they're kind of obvious. Okay. All right, so that was exam number 1. So again, a lot of times, if you've never had a class with me before, half of doing well in my class is learning how the questions are going to be asked. And then when you study for the next one, you'll have a better idea how to study because you know how I'm going to be asking questions. And these last 4 questions, I try to give you a bunch of practice examples in the in Moodle there. But the whole point is come up with a hypothesis. It has to be an educated guess, right? It has to be based on the facts that you learned in class. But, half the time I don't know the answer because there is no answer. It's just, come up with a hypothesis. What do you think it is that's doing this? Okay. Any questions or comments about the exam? Will bonus points be added? Yeah, the thing about the bonus points is, in the old days on a paper exam, I could just put them on top of the exam and here you go. With Moodle, I can't really do that. So I just put them in a column by themselves. So then I'll add up all your exams and then throw the bonus points in on top of it and divide by 4, right? Or actually 6, because we'll have 4 exams and a double counted final exam. So yes, that's a long way of saying yes, Abigail. Sorry. Any other questions? So the grade that's listed in Moodle does not include the bonus points. Correct. So take whatever's in your bonus thing and then add it to that exam if you want to do that. Do it that way. Okay. Any other questions? Unless, of course, I don't know if other students have told you this, but if I don't like you personally, I just delete your bonus points at the end of the semester. It happens. It's been known to happen. Oh, go on. I like all you guys. All right, so let's rock on, people. We are going to start the next chapter, which is chapter 12. Okay. Get myself set up here. Get myself prepared to talk about these important topics. Okay. Again, it's like I prepared enough today. I prepared today the same amount of material I would do if we were in class, but things tend to go a lot faster here because I'm not writing on the board. So this might be a little bit of a short lecture, unfortunately. I know how you hate short lectures, but here we are. Just preparing my real estate. Okay, the endomembrane system. Now, this is something I never personally did any research on, so I don't have any hands-on experience with this, but it's kind of an interesting chapter in that it explains how proteins are made in the cell, more so than just translation. So, and by proteins, I mean mainly membrane and secreted protein. So a lot of this chapter is about how those proteins get made and how they get put where they need to go. So that's rough ER and Golgi. But then you also have your smooth endoplasmic reticulum, which has got a lot of roles. We've seen them already, some of them in calcium storage. And then you got your endosomes and lysosomes, which is about endocytosis. So you pull something in and then you got to destroy it, right? Break it down and use it for food, or just break it down to neutralize it if it's like a bacterium or something. And then peroxisomes, which I'm not sure we're going to talk about. We may or may not. I'm not a big fan of the peroxisome because I don't really have a good grasp of what it does. So I hate to read it from the book and then tell you what the book said. That's kind of a waste of time. And then transition vesicles. So just vesicles floating around in the cell, not floating around, moving around in the cell. Okay. Here we are. Here's your rough ER. And notice that the rough ER is contiguous with this outer membrane of the nucleus. Okay, so remember your nuclear envelope is two membranes, an inner and an outer membrane. This outer membrane then becomes contiguous with this rough ER membrane. And it's mostly flattened sacs. You've got a little bit of tubularness over here, mostly flattened sacs, and rough because it's got ribosomes on it. Okay, and they're not permanently attached there. We'll see how they come off and come back on when needed. And really close to that, usually associated closely with it, is your Golgi apparatus. So proteins are going to be made in the rough ER and then sent via these transition vesicles over to the Golgi apparatus. And the Golgi will then process the proteins that have just been made. So doing post-translational modifications of the proteins. And then over here, contiguous with the rough ER is the smooth ER. Smooth because it doesn't have any ribosomes on it. And it's kind of tubular, right? Compared to these flattened sacs over here, it's a very tubular kind of a thing. And tubular because you get more surface area. And a lot of what happens with smooth ER is a surface phenomenon. So it's very, very much like that. Okay, and then over here are again transport vesicles coming off the other side of the Golgi. And these are endosomes. And the endosomes have the enzymes for lysosomes inside of them, but they're not activated yet. They're sitting there waiting. When a vesicle is endocytosed with something inside of it, a food particle or a bacterium or something, it'll then fuse with these early endosomes and become late endosomes. And once they're late endosomes, it'll activate that protein pump we talked about in the last chapter, the V-type pump, and it will become acidified, and now it's a lysosome. And the lysosome will then break down whatever is inside of it and then sort it out, whether it needs to go this way or that way, or even spit it back out of the cell. The lysosome will work with that. Here's your dreaded peroxisome over here. So the peroxisomes are basically making peroxide, which is a free radical type thing. And I think it's involved in either breaking down fats or neutralizing free radicals in the cell. You know, free radicals are really dangerous because they react with anything they run into. So they destroy a lot of things in the cell. So those are your peroxisomes. Okay, and here is your rough ER. You can see how different rough is from smooth. Both the fact that it's got all these dots on it, which are ribosomes, and it's like sacs like this, versus your smooth, which is tubes and doesn't have the little dots on it. So that's your rough versus smooth. Okay, so first thing we're going to talk about is the rough ER. What's the important thing about the rough ER? Now, when we were in high school, back when we were in high school, pretty much they say, what's the rough ER? It makes proteins. Sort of, yes. But it's not going to make all proteins. If a protein is soluble and it's meant to be inside the cytoplasm, it's not going to be made by the rough ER. It's going to be made just by free-floating ribosomes in the cytoplasm. I'm not sure if they're free-floating. They might be attached to cytoskeleton somewhere. But basically, cytoplasmic ribosomes are going to make those proteins. The only time you need a protein to be made in the rough ER is if it's going to need to be inserted into a membrane. So if it's a plasma membrane protein or any other protein that's in a membrane in the cell, or if it's going to be secreted from the cell. In terms of the membrane proteins, as you probably know, at least I hope you knew, if you've had biochemistry, membrane proteins have got transmembrane regions. Okay, so these are regions that go through that lipid environment of the membrane. And so because they're in the lipid environment, they've got all these non-polar side chain amino acids there. Isoleucine is like 3 carbons or something like that. Phenylalanine's a phenyl group. Valine, alanine, valine, isoleucine, tryptophan, tryptophan? Tryptophan. Isoleucine, leucine, leucine, isoleucine. So these are all very non-polar. So these amino acids would not be energetically favorable outside the cell, which is hydrophilic, or inside the cell, which is hydrophilic. Okay, so if you made this, and these amino acids are hydrophilic, so they are perfectly comfortable outside the cell. These are fine in the cytoplasm. Now, if you took this protein and got rid of the membrane and just let the protein in a water solution, hydrophilic solution, it would curl all up funny. And it's curling up to hide those hydrophobic amino acids because they don't want to be exposed to the water. So it's going to fold up wrong, and it's not going to be functional because it's trying to, or it needs to get the hydrophobic amino acids away from the water solution. And this was a struggle for a long time. You know, when people try to figure out the structures of proteins, it's easy enough to find out the amino acid sequence, and you can, by molecular biological means, isolating the messenger RNA, making cDNA, sequencing to cDNA, and you can figure out that primary sequence. But then what does it actually look like in 3 dimensions? If it's a soluble protein, all you have to do is dry the water out basically and make a crystal out of it and do X-ray crystallography. If it's a membrane protein, you got all this membrane in the way. You can't do X-ray crystallography with membranes. At least back in the day, you couldn't. So how do you get an X-ray crystallographic look at a protein that can't be made a crystal of? So it was really hard. Anyway, that's neither here nor there. Bottom line is the reason why we're making these in the rough ER is because this segment here, this transmembrane sequence, has to be put in a membrane as it's translated, co-translational insertion. Otherwise, it's going to fold up crummy, and then the protein won't work. Secreted proteins, hang on a second, let me look at something. I have a figure on this that I do not. Okay, well, let's look at this one. Okay, secreted protein. So if you make say you've got a hormone like enkephalin, okay? It's an opioid agonist. It's a protein of about 90 amino acids. So something like that, if you made it in the cytoplasm, it would be easy to make in the cytoplasm because it's soluble. You couldn't get it out of the cell. How do you get something that big through a membrane? You really can't. Remember, we said in the last chapter, the biggest thing that can get through a membrane, diffuse through a membrane, is like something the size of glucose. So this is huge compared to that. And you really can't get a protein transporter to get it out. It's just too big. How do you get it out? The only way to secrete a large protein like that is to put it on the other side of the membrane as it's made. So again, co-translational insertion. As the thing's being translated, is being put across the membrane of the rough ER. So then it gets passed along inside a membrane in these transition vesicles over to the Golgi. Now it's inside the Golgi, passing from sac to sac of the Golgi. It's inside the Golgi. Now these vesicles come over and the membrane of the vesicle fuses with the plasma membrane, and now it's outside, okay? So the only way to secrete a protein then, again, is to put it on the other side of the membrane as it's made, and then you're always using membrane vesicles to move it around and using that to exocytose it. Okay.
Sorry, so just to be clear, the blue dots are proteins in this case? Like the blue dots within everything?
Right, exactly.
Okay, I just wanted to make sure.
Yeah. Okay, so that's the why. So rough ER, membrane proteins, and secreted proteins. That's Golgi. That's my old electron. Okay, so how do things get made then? The next question is how do you make something on the other side of a membrane like that? And that's where co-translational insertion comes in. Hang on a second. Look at my notes real quick so I don't forget something. Okay, so these are going to start their translational lives in the cytoplasm like every other protein. It starts to get translated there. So the ribosome grabs the messenger RNA and then starts to translate. Now the first 6 to 10 amino acids, and frankly, I made those numbers up, but the first few amino acids are going to be called a signal peptide, so a signal sequence. So this is something that's not going to end up in the final protein, but it's important to get this complex over to the rough ER. So your first few amino acids then are the signal sequence. This, when it gets exposed, is going to be recognized by the signal recognition peptide. So this is a protein that will then bind that ER signal sequence and stop translation. So translation stops here. Now it's going to bring this over to the rough ER. And the rough ER has got these pores in it. This is how things are going to get into the rough ERs through these pores. Now the SRP has got a receptor for it right next to the pore. So this SRP receptor is attached to the pore, and you can see this little nib sticking out here binds to that. Now you've got the whole receptor mRNA complex SRP attached to the pore. Now it's going to take some energy to get that released. So we pick up a couple GTP, and that displaces the signal peptide. So now it comes over into the pore of that channel protein, and it binds to the pore and gets stuck there. Now, GTP then will be hydrolyzed, and that energy will allow the SRP to leave. Once the SRP leaves, then translation can proceed. So translation starts to happen, and as it's happening, that signal peptide stays here attached to the pore, and the peptide comes down into the lumen of the ER. Okay? And when enough of that protein comes through, then this enzyme here, signal peptidase, will cut the signal peptide off. And that peptide will go into the cytoplasm and be degraded and amino acids reused. And then translation will continue, protein coming in, and then translation ends and the carboxyl terminus comes into the lumen. So now the whole protein is in the lumen of the ER. Okay. I think it's kind of a cool mechanism. But that's how it gets into the ER. Okay, so that's for secreted proteins. And again, we put it inside the lumen of the ER, and always think inside the lumen of the ER is like outside the cell, because when that fuses, it'll be extracellular. If something is in the cytoplasm, it'll always be in the cytoplasm. So the next question is then, how do we get membrane proteins into the membrane? And one way to do, we got two different types of proteins we need to think about. Well, three different types of proteins we need to think about. One is a protein that's got an extracellular amino terminus. One is got an intracellular amino terminus, and the other is multi-pass proteins that go through the membrane multiple times. So we've really only got three things we need to do all of those proteins. And the first we've seen already, signal peptide. So if you've got a protein that's going to have an extracellular amino terminus, okay, which means it'll be inside the lumen of the ER, it starts out the same as making a secreted protein. The first steps are exactly the same. The mRNA starts to get translated, SRP grabs that signal peptide and brings the whole thing over to the pore in the rough ER. And then that signal peptide gets released and gets put into the pore, it sticks here, and now translation starts and the protein starts coming down into the cell, into the lumen of the ER. signal peptidase cuts off the signal peptide and that goes away. Now, what's different is as it's coming down through, it's going to hit a stop transfer sequence. What is a stop transfer sequence? It's basically 18 to 22 hydrophobic amino acids. This is going to be a transmembrane region in this protein eventually. So when that very hydrophobic thing comes through the pore, it hits the middle of the pore and it gets stuck there because it's hydrophobic. It will then get passed out of the pore and into the membrane. And once that happens, then translation can proceed. And here, all the translation is in the cytoplasm. So your carboxyl terminus is cytoplasmic and the amino terminus is extracellular. Okay, so stop transfer sequence. The other question is, how do you make a protein where the amino terminus is intracellular instead of extracellular? Well, for that, we don't have a signal peptide because a signal peptide will always put the N terminus inside the lumen. So what happens here is you've got an internal start transfer sequence. So this is, again, 18 to 22 amino acids that are hydrophobic. So translation starts in the cytoplasm, and the first whatever amino acids are all going to be hydrophobic, so hydrophilic, so nothing really happens. But then a stretch of hydrophobic amino acids come out, and the SRP grabs that. And SRP then grabs the whole complex down to the pore and the rough ER. And here's the beginning of the protein, right? The hydrophilic amino acids that began the protein. And then it hit this hydrophobic stretch. SRP grabs it, brings it over here. It binds inside the pore, and then translation can proceed. And it goes and goes with the rest of the protein going inside the lumen of the ER until you get to the end of the protein. And now you've got your start sequence stuck here. It gets transferred out, and now you got a one TMR protein with an NH3 terminal in the cytoplasm and a CO terminus in the lumen of the ER. And again, remember, this will be extracellular once the protein's put into the plasma membrane, and whatever is in the cytoplasm will stay in the cytoplasm. Okay? So that is your single pass transmembrane with an intracellular N terminus. And then the third type is a multipass protein. So how a multipass protein is put together depends on whether it's going to have an intracellular or extracellular amino terminus. If it's going to have an extracellular amino terminus, it'll start with a signal peptide. If it has an intracellular NH3 terminus, it won't start with that. It will start with a start transfer sequence. Now, what makes this multipass? So up to this point, it's exactly the same as the previous slide, okay? Cytoplasmic NH3, and then a start transfer gets stuck in the pore. Now, what's different is as this is going through, it's going to come across an internal stop transfer sequence, okay? Which is, again, 18 to 22 hydrophobic amino acids. When that gets into the pore, it stops. And then now you've got two hydrophobic regions. They get transported out into the membrane, and then translation continues. Now, if that's the end of the hydrophobic stretches, then you'll end up with a carboxyl terminus here in the cytoplasm, as well as the NH2 terminus. But in this particular protein, along the way, we end up with another start transfer sequence and then another stop transfer sequence. So this protein so far is going to have 4 transmembrane regions. Excuse me. So basically, it's the translation is going to stop. The passage through the pore is going to stop every time it hits one of these sequences, and then that becomes a new transmembrane region. So I made an animation about this years ago, but I made it with Flash, so I can't put it on the web anymore. The web doesn't do Flash. But I can still do it on my computer, I think. So this is the Flash program Animate, which is what I made it with. Play. Play, damn your eyes. This may or may not work. We'll see. Okay, I can't stop it, but you can watch it go. See, there's your peptide sequence. Now here's your stop transfer. Here's a start transfer, stop transfer, start transfer, stop transfer, and there, 5 membrane regions with an extracellular amino and intracellular carboxyl terminus. Can I do this again? See, none of the actions are going to work. Let me do it again real quick. None of the actions are going to work because it's no longer supported. Okay, so signal peptide, cleave it, stop transfer, no, start transfer, sorry, stop transfer. I don't know, they're all the same really in the end. And scene. Okay. I'm going to see if I can do some kind of like a screen grab so I can just take all my old animations and just do videos out of them because I can't put them on the web anymore, as I've said. Okay. Any questions about that? Oh, Claire, you're muted.
I didn't say anything.
Were you saying something, Claire? Or Carrie? Sorry, Carrie. Yeah, you're still muted. You're muted again. There you go.
Okay.
Good.
Did you said there were 5 membrane sections? Is that what you said?
Membrane transmembrane region, TMR.
Transmembrane
region.
Okay,
yeah.
That's what the TMR is. Okay, great.
Yeah.
Thank you.
Any other questions or comments here? Okay. All right, so that's all I have prepared for today. Typically that lecture would take me an hour and 15 minutes, but you know, on this this platform it's a lot faster. I hate to go too fast and give you more information than everyone else got because that might make your exams harder. I know you're not a fan of hard exams. Anyone have any questions for me, comments? When is our next exam? Do we have another exam or are we done?
Says on Moodle it's March 23rd.
Oh my God. That's like 3 weeks.
Yeah, please don't move it up. It's good right where it is.
All right, well, I'll leave it where it is.
I think on the syllabus it says March 16th.
Oh,
That's
no.
better. That's
I think Moodle has a different date though.
Oh,
Let's change Moodle.
no, we don't like that.
Oh, it's just that, you know, I've got about 2 or 3 more lectures and then I got nothing else to talk about for this exam. I could come up with new stuff. We could maybe play some games. I don't know.
Hey.
I'm sure I could come up with stuff.
I don't think I trust you. We can call it spring break.
Hey,
That's a good.
hey, that's not fair.
We need to talk, by the way. You're applying to school this year?
Yes, we do need to talk. I was thinking about that. So yes,
Okay.
we will talk.
I'll send you a Zoom link.
Okay.
Tyler, is Tyler here? Tyler Montana? Is there a Tyler here? He's a bit of a lurker. I guess he's not here. So he's working on the next exam game, like the last exam game. I think it's almost ready, so I can get that up for you pretty soon too. All right. Well, I'd like to sit here and just keep talking to you, but I really don't have anything else to talk about today. Any comments or questions? Nothing? Nothing? Okay,
So you're going to decide when the test is going to be, or you're going to leave it at the 23rd? Like, are we going to potentially move it up?
I think it's going to have to be the 16th because
Okay.
what's today? The 4th?
Yeah.
Yeah.
So next week will be the 11th. So it'll be the week after that. I mean, that's about when it's going to have to be. Yeah. Otherwise, I got nothing to talk about for a whole
All
week.
right. Sounds good.
Okay. All right, guys. Have a good weekend. We'll see you on Tuesday.